Hi everyone, welcome to today's episode. My name is Ikrame Housni. I'm a master's student in biomedical engineering and part of the QBIN Scientific Student Committee. And today I'm very excited to introduce Dr. Emily Coffey, an associate professor at Concordia University and the recent recipient of this year's Rising Star in Bioimaging in Quebec award. Dr. Coffey has a strong academic background with an honors in psychology from the University of Ottawa, a master's in brain and cognitive science from the University of Amsterdam, a PhD in neuroscience from McGill University at the IPN, and a postdoc that she did in Germany. She leads the Coffey Lab, which focuses on how music and sleep can enhance brain function. Her team uses advanced neuroimaging to study auditory processing and the benefits of musical training, as well as exploring how sleep interventions can improve memory and quality of life. So to start, maybe can you start by describing your research career path and what drew you to specialize in your current field of interest?
Okay. So my research career path has been a little bit rambling. I started off looking at pilots and workload because I was a pilot at the time and I was interested in why people crash and what happens in older adulthood, in terms of how people perform and why they miss certain indices that could cause safety problems. So I was at the University of Ottawa at the time, and there was a lab at Carleton University, which is still there, with Dr. Chris Herdman, and they were interested in that aspect. So I was building a flight simulator and see when older adults missed things that could be safety relevant. So that was step one. After that point, I got a job at the European Space Agency because they had a human behavior and performance group, and they have a young graduate trainee option, which is like a job for a new graduate, as the name suggests, which actually makes you work in the space agency with the experts. And in my case, I was working with people who train astronauts, and they were interested in having someone who has a psychology background, but also has some operational experience. And because I was a pilot, I had both of those, and it was a good fit. So I moved to Germany then. And after about a year, I really enjoyed that and learned a ton. But I realized that within the space agency, there's not too much more you can do with that aside from astronaut training, which was really cool. But I wanted to go back to science. So I did a master's in brain and cognitive sciences at Amsterdam University.
Okay.
And that was great because it really exposed me to a wide range of different techniques in cognitive science, and in kind of the more, I don't know, hard end of psychology, if you will. I really enjoyed that. And during that time, I kind of did two theses. One of them was again about workload, looking at combining functional near-infrared spectroscopy and EEG to measure people's workload when they were doing some basic tasks. And then the other one was looking at appetitive conditioning in mice in the visual system, so something completely different. And I just didn't really know what I wanted to do, so I was exploring a few different areas. And that made pretty microscopy images essentially. And after that, I decided I did want to continue research, and I moved to McGill, and I ended up with Dr. Robert Zatorre and studying music and neuroplasticity. And from there, it's been very auditory, but now I've gone also to sleep research to see how those two combine. And in the work that I'll present at the QBIN, because I have these kind of three different areas, workload, audition, going from very basic processes up to neuroplasticity and complex tasks, and then sleep. I'm gonna pick one of them essentially, because otherwise it would just be too confusing. So I'm
Sure.
mostly going to talk about sleep and its interaction with sound.
Oh my God.
Okay,
Okay.
that's very interesting. I always um, enjoy listening to PIs or academics and senior researchers explain how they got to what they're doing right now today and the current research of interest, because it's hard to imagine how you're going to such a niche specific research subject. So, for example, one of the many things you're looking at is the relationship of sleep with sound, and it started with you being a pilot and then having an interest for how those pilots were reacting, and one thing led to another, and today you're an associate professor.
So you've been in academia for quite a while now, and you have your own lab.
Mhm.
Um,
is there any part of your work or any study or any publication that you think stands out to you and is like favorite?
I was thinking about that because he did send me the questions in advance.
Yeah.
And I think what I would choose for that is the cortical contributions paper in Nature Communications in 2016. And the reason is because it actually did have a fairly big impact within our subfields, but more so because of its generation. This was a small seed project that I decided to look at with my professor a little bit randomly after reading a couple papers to show that it is possible actually to look at some of these early auditory responses using magnetoencephalography or MEG. And we had just gotten a MEG at that time at McGill, and Sylvain Baillet was there and was setting up the program, and they were trying to encourage people to use it. And I said, oh, I'll apply for some seed funding for this and see if I can get this project going. And we discovered that a small auditory signal called the frequency following response, which was thought to come only from the brainstem, is actually also coming from the auditory cortex. But to get there, and that was important for a number of reasons, just to understand what that signal means, because people use it for all kinds of ways of looking at things like aging and differences between populations and what it means for neural integrity and all this stuff. So it sort of changes the interpretation of a lot of previous results. But I think what was fun for me is really just following what the data were telling us and thinking of creative ways to get at this new signal that I was unfamiliar with and that other people hadn't really gone too deep into the brain by that point to look at how much you can get out of a MEG signal. So working with Sylvain Baillet and other experts and fiddling around by myself, we've managed to come up with 6 different ways to show it was true, because basically no one believed us. And those all sort of came together. And now the study has been replicated by several other groups. And we can see that it's actually is contributing to development in that area. And I feel like I actually had some kind of an impact in that area. So
Oh, wow.
happy about that.
Yeah, I don't think that's an impression. That's really interesting. And it was initially just an explorative study. You were not going with any like hypothesis or anything. You wanted to see, or did you have an idea on it and you wanted to?
Yeah, I mean, our idea was, can you separate the sources? I knew that from measuring frequency following response from different electrodes across the brain, I was getting all kinds of stuff that didn't really make sense.
Yeah.
If it was only coming from the brainstem. So I didn't yet have the notion that maybe it's coming from the cortex as well, but I did think, okay, maybe we can separate some of these sources out. And in fact, it worked much better than I expected in the end.
Oh, okay. And because MEG is known to have, I guess, compared to MRI, lower spatial resolution. How were you able to separate those sources?
Well, I guess a lot of it relies on the more advanced source reconstruction methods that have been evolving in the last few years, but not too many people have applied them to deep sources. So there were some hints that it was possible to kind of trace a little dipole as a signal is coming up from the auditory nerve through the cochlear nucleus and the inferior colliculus and the thalamus and then to the cortex. But for more advanced sorts of analysis, you really need to do the full source reconstruction using the subject's own anatomy. So I did a lot of playing with that. And of course, as you get further away from the sensors, the problem becomes less well constrained and you get sort of blurring of the signal. So a big part of what we were looking at there is seeing how well can you separate out those signals. And you can use little tricks like the delay between a signal coming from one spot to another. Or we did some kind of forward projection to say, okay, if the signal were coming entirely from this source, how would that look on the sensors? So we got at this from all different ways, and we also increased our signal to noise ratio as much as we could. So if you're studying an auditory evoked response, like just a normal one, maybe you need 100 or 200 repetitions of the sound that you're interested in hearing or interested in looking at the response to. If you're looking at frequency following response, which is like a very high frequency evoked response potential that encodes some information about pitch. Should have probably said that earlier. Then if you do that, you normally in each, you need 2,000 iterations. So that means someone gets to sit there and hear da, 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 da for something like 15 minutes. Now in MEG, we didn't know if we could even see the signal. We knew that it was going to be quite deep and quite hard to see. Yeah. So we really overdid it. So we went with 12,000 dot, like a full hour of listening to da,
Oh,
and
wow.
then we see the signal. And we also reduced it a bit afterwards, and you can see it with 6,000 and a little bit with 2,000. But the point is that you can go deep with MEG,
Yeah.
but with all physiological signals, it's a matter of signal to noise ratio. So you need to change something, and that something can be finer use of these reconstruction techniques using anatomy, but it can also be increasing your signal to noise ratio through experimental design.
Okay, so you really did reach that result through different methods by applying different methods on a very on a theory that's really cool, because there's like these very impactful researches that come up, and it's really interesting to see the progress of those findings and how much was like really learning and deciding the path in which you want to take it as you're learning about it and as you're doing it, as opposed to just having a clear picture from the start. So that's
Yeah, definitely not. I'm definitely not doing the kind of research where you necessarily have a clear picture from the start. I think it's probably important to do for things like clinical trials and such, where you want everything set in advance and you get a yes, no kind of answer. But in the kind of work we do with neuroimaging, there's a lot more exploration. So you also have to be careful not to delude yourself, and to make sure that when you think you have an answer for why it's happening, you can then sort of triangulate it and try to do some analyses from different perspectives and make sure that everything's converging.
Yeah.
And only then you should be reasonably sure that it's a real result and not something that you're making up because of wishful thinking.
That's very true, because it's really easy to be biased. That's a very valid. And do you think this research influenced how you now approach research?
Certainly. I think that's the kind of research that I want to be doing. When, yes, you have quite detailed methods, and you're really using the resources at the edge of what other people have shown that they can do, but I'm not necessarily only working on methods development. I'd like to be able to use them to better understand human cognition. So I'm always kind of playing with that interface of, we're developing some tools, or we're at least exploring how they can be used in new contexts, but we want to say something about the brain. We're not just looking at the tools.
Yeah, for sure. But then it's also it's kind of, I feel like the methods part of research is maybe the gratification from it is maybe not the same as you would get from just answering questions about the brain. But the more time we spend in like fine-tuning the methods and making sure that the result, the way they're used, is how they're meant to be used, and that we can actually trust them and that they're reliable in giving us the results, it makes it then easier for when you start to study the brain. But then again, you always have to go trials and errors, so you there's no escaping going back and forth.
Yes, and especially since other people using these methods are typically not using them for the same reason that you might want to. So even if they've been validated, you still have to kind of keep an eye on that and do your own validation, or figure out, or just be aware that there can be things that aren't true that you need to figure out multiple ways of getting at.
Yeah. No, that makes sense. And I guess on a more personal level, what are the challenges in research that you found, I guess, harder to overcome?
You mean in research itself, or in
in research, in a career as a researcher, or potentially as a researcher itself? Feel free to answer that question, actually, how you understood it. Yeah.
Okay, maybe I'll start with research itself. I mean, it's just very uncertain. So things can go well, or you can have major setbacks, and so you have to be sort of psychologically resilient and willing to go back and spend another 3 months looking at something, which can be hard. We're dealing with a situation in our lab at the moment that might be the case. So keeping everyone's spirits up, including my own, can be a challenge when that happens. But I think maybe career-wise, there are a whole lot of other factors which make it challenging. Of course, balancing teaching and research and service jobs and all the administrative stuff we have to do, and going out and finding funding and worrying about whether or not your students have enough funding and all this stuff. That's a whole other game one has to do, as well as doing research.
Yeah, that's very true, because we often think as like
career as a PI is that you can just do your research, you can do your own questions, which are definitely the perks of being a PI, because you can choose the direction of your findings, of like your research and interests. Although there is some pressure depending on what reviewers will want to publish, of course. But another very, very, like, I would say it's almost, from what the PIs I've talked to about tell me, it's about like 50-50 between research and grants, and making sure you have fundings to do the research that you want to do. And like there's always conversations about like PIs are spending too many months writing grants, and they happen at different times of the year. And you, even if, because you don't know when you're going to receive a response for a grant, you have to still apply because you do not know the answer. So it's a bit burdensome. It's the part of a research career as a PI that I think that it's the one consensus you can find between PIs. It's that it's a bit draining.
Yeah, I think I like writing grants. With the caveats, several caveats. I mean, grant writing is a chance to step back from the research that you're actually doing and plan out your future and think about the next questions and read the literature and put everything together. And often when you go through that process, even if you don't get the grant, you will use those ideas in the future, provided you get money eventually from somewhere else to do it. What's not fun about the grant is the pressure and the stress of what happens if I don't get the grant. Can I feed my graduate students and that kind of thing? So I think certainly if grant success rates were higher, it would be less unpleasant. But for something like CIHR, with the funding rate is 13 to 15%, even for established researchers, it's pretty hard to spend a month of your life writing a grant and then have it slightly rejected. Like you didn't quite make the cutoff, and then you have to wait another 6 months or a year before you have a chance of doing it again.
Yeah, that's true. And apparently after the new regulations with the CHR, there's going to be even fewer people that get selected for grants, because they increase the pay for the PIs to be able to pay their students after the inflation, which makes a lot of sense because there needs to be adjustments, but it's the funding doesn't seem to change to just keep the same numbers of PIs being granted funds every year. So I feel like it's not necessarily going better.
Yeah, I think we're all holding our breath to see how that's going to pan out. Certainly, it's nice that the student scholarships are finally being increased. We're all very much behind that. I don't know how it's going to settle out on the grant side. We'll have to see if
I think it's questions that we're going to be able to answer as like more of as the grants are being given out, and that we see how it evolves through the years. But that makes a lot of sense. And a question that could be, you can take it from a philosophical standpoint, you can take it as you want, but are there any issues in your field today that you find particularly contentious?
Guess I would have to pick a field first. I don't think in particular. I don't know if I have a good answer to that one. Certainly, there are lots of little frontiers where people are debating back and forth what's true or what's not true, or how should we do this. Maybe one of them in neuroimaging in general, just in case it's interesting for students in this area, is the debate about whether or not we should have bigger and bigger sample sizes. So if you ask the statisticians, it's always, oh, you need 200 brain images to do whatever it is that you want to do. But that's often out of alignment with funding and with resources, essentially time of students and ability to analyze and all this. So I think there are many examples from animal research and from other types of research where you're increasing, again, your signal to noise ratio by collecting more epochs or collecting longitudinal data, or there are other ways to get at problems, or even to, as I was saying before, sort of triangulate your problem from several different directions. So practically speaking, let's say you're doing a functional magnetic resonance imaging study, and you can also collect some diffusion data, which is easy enough to do if you can put it in your protocol. Let's say you find something in your fMRI, maybe that's exploratory, maybe that you expected, and you start to develop an idea that maybe two brain regions are talking to each other more than you thought before or something. You can also go and do a different analysis on a relatively independent aspect of this data set by, say, going into the diffusion data and seeing how well the strength of diffusion parameters are correlated with some behavioral score.
Yeah.
So that's a way that by putting multiple types of data together or multiple trials, like what I was saying with the 12,000 DAWs for my FFR analysis, in that case, you maybe can have a smaller sample size. And then that's comes back to using our collective research resources more efficiently and kind of more responsibly. So I think the field has to reckon with that a little bit, because when you send stuff into review with 10 subjects, they're just going to say, no, you can't publish that, even if each of your subjects has millions of points of data.
Yeah. Then that's a problem that I feel like we hear a lot about in research, the whole sample size, that or the p-value, but the whole statistics kind of not caveat, but a harder side to fulfill in research, because some data sets are just very hard to have in like high numbers. Or if you do have them in high numbers, the quality isn't necessarily as good, because you have to merge many multi-sites data sets, and just changing the scanner will definitely change your data. So there's a lot of definitely a lot of things to unpack there. And I think there are some standards in research that have been set for so long, and that it's just maybe hard to change things necessarily now. But I feel like it's going to be, if the change is warranted, it's going to be gradual, and hopefully, you never know.
Yeah.
Again, which from your point of view, which aspects of research you find most gratifying in your own research, in your own day-to-day? Which part do you feel is most rewarding?
Actually doing research. So there are really a lot of other duties when you're a PI, and some of them you might like more than others. But I think many of us who get into research because we enjoy that, we really treasure and value those small amounts of time that are left to us to actually pursue our own research or work with our students on our joint research, and help to mentor and guide them as they develop the skills they need. I really like to sit down with my students and look at little plots of data and try to understand what's going on and think about what we do next, or just design studies and things. I think that's why we do this stuff, and that's the fun part.
Yeah. Would you do you think the system that is now in place is like inevitable, kind of, or do you think that we can actually change easily a few things if, let's say, the infrastructure you can say in the infrastructure of research, or even at the university levels, that could allow PIs and students to just focus more on the research?
Ooh, you're going to get me going on this one. So a big problem, I think, in recent years is that the universities have tried to cut costs by putting more and more administration on researchers. And in part, they've justified this by saying, look, we have all these fancy online tools. You can submit your own expense reports and you can do this and that. But it becomes a huge administrative burden. And there's often no one who's expert enough to help you with those systems. So everything is inefficient. It takes time. So I would really like to see universities reinvest in competent administrative help that stays around for a long time, so that we can spend less time doing things that you wouldn't really think that professors should do, and we're really not good at necessarily, like understanding the legal implications of getting someone's work permit from Singapore or something like that. That can take a huge amount of time. I think you need to preserve the time of the researchers to work on the research and with our students. And if we do that, the quality and the quantity of research is going to go up just with the same people. You don't really need to do anything else.
Yeah. So would you think a setting where every lab has a person that's responsible for all the administrative and financial components of research, do you think that would be an investment that would come at a cost? Probably. But do you think that would be a worthy investment for the quality of the research and the amount of the research that can be produced?
Absolutely, but it doesn't necessarily have to be one person per one lab. I think you could have a few of them for the department that are assigned to several labs. That's probably kind of the right amount of work. I do think it's really helpful to have senior people in the lab that are there long term. And right now, there's almost no provision for doing that within the Canadian funding scheme. So what I mean by that is people who are not necessarily going to go on to be professors because they don't want to, or because maybe they're not interested or don't have the full skill set that includes teaching and all that, but they're very good at research and at teaching people within the lab and understanding the technical details. And right now, bigger, well-funded labs can typically keep a research associate on for a longer period of time, but it's not something that many labs can have. And that's a huge benefit for training people and for increasing the quantity and the quality of research output. The flip side of that is there just aren't enough jobs for our graduates. So we're training a bunch of PhDs, and yet there's only a few PI positions that there will ever be, essentially. So we're then sending people off into industry and such and saying, look, you can maybe transfer some of these skills to commerce or something. Whereas I think it would make a lot more sense to maybe train a few fewer people, but figure out a means of retaining them for an actual career in research that's not just being a professor.
I see how that would be very helpful. And the research assistant can also communicate to the responsible for the few individuals that are responsible for the financial and academic.
Yes, they could help with that.
Because the reason why I think that absolutely definitely from a research standpoint, but as well as like dealing with the kind of the rest of the stuff that are needed to run a lab is because a lot of things, even if they don't take much of your time, they will take a lot of your mental effort and like mental space, and just going back and forth in emails with the responsible, like just for example, I was recently helping my supervisor to just fill out some financial forms, and just doing that had a lot of back and forth in email, and it does take mental space, and it does. So I think definitely if there can be an investment and research assistants that kind of really help, like kind of keep the backbone of the lab outside of like the research part and with the research part, that would definitely be ideal. I don't know how feasible it will be, especially not in the near future. But yeah. So my one of the things that I'm curious about is if you had anyone in your career, it can be someone professional or personal, but someone that you think was a mentor to you and had a big influence on you becoming a researcher or being the researcher that you are today.
Yeah, I guess for me, it's an obvious one, my PhD mentor, Dr. Robert Zatorre. So he's been a lovely person to work with, and I think he's really influenced how I like to do science. And he showed me as well, in science, as in any human endeavor, you do get competition and politics and all this stuff. And I think he's very much stayed away from that and shown how you can be an excellent researcher and enjoy yourself doing science whilst being a nice person and treating people extremely well. And I think that's an excellent model for any scientist to take on.
Yeah, I do understand the importance of this, because it's true that in research it is not in research it is such a competitive field, and it really is kind of like survival of the fittest, whether it's with funding or with the research amount that you have to do, and not just the quality but just the quantity of the research itself. It can be very difficult and draining. It's really easy for the career of a researcher to take a bit kind of I don't want to say sour taste, but just it can be just less entertaining.
Yeah, you need a balance to that. And I think the balance for people like me and I are that you are working on interesting problems and it's at least enjoyable most of the time. And you can work with friends or like-minded people who become close colleagues and friends. And then you have these little moments where you're like, oh, we're seeing something for the first time. No one else knows this. And you get a little tingly feeling and you have people to share that with. Or you sit around and you have a nice dinner and you come up with some fun ideas of new research directions on the back of a napkin. And those are the kind of things that I think really keeps me going and that I find are most of the time an adequate balance to the various stressors of fighting with administration and things like that.
The research on the back of a napkin, I mean, I've seen it in movies, but the passion is real for sure. And are there any, you mentioned that there were many, like there's this possibility of when you're in formation to becoming a researcher, you have colleagues and that eventually you meet again and you discuss your findings and perhaps have collaborations with. Are you still in contact? Do you still know or hear of the researchers of many of the people that were with you probably in your PhD?
Yes, I mean, some of them have taken quite different paths. Like one of my closest friends during my PhD has become a clinician and moved back to where she came from in Germany, and has a family now. So I mean, we talk to each other from time to time and go visit from time to time. But I don't think there are too many people have ended up exactly in my area. But you know, people diverge, and then you get new ones, and it all kind of there are some postdocs and stuff that I continue to work with. Who are now professors in different places.
That's really cool. That's really cool. And also, I think the conferences, there are many neuroimaging conferences that kind of allow you. There are some people that you just have like a yearly connection with. Like you're going every year, you're gonna enjoy meeting them, and yeah, it's definitely the kind of collaborative family aspect of research, I guess.
Yeah, that's the fun part of conferences, and especially doing them face to face and not over Zoom, is seeing your friends once in a while.
Yeah, especially after COVID, the Zoom part, yeah.
Yes,
sure.
agree.
And throughout your career in scientific research, specifically as a woman in research, have you encountered any specific challenges that you think would perhaps be specific to you being a woman?
I would like to say no. And I think in my earlier years, I felt quite protected by the professors that I was working with and the environments that were there. I think more recently as a professor, I have noticed some instances in which you, as a woman, may feel that you have to earn the respect of those that you're working with or teaching, or more so than a man would have to do. I'll give you an example in the classroom. If I walk into a classroom and say, "Hi, hey, my name is Emily, and I'm your prof," because I'm a relatively young woman in the professor side of things anyway. I definitely feel like I have to prove myself and my knowledge and justify why I'm there and why I should be telling people things. And there's more pushback for things than marks that other professors report. Whereas, and so I take a few steps for that. I'll ask them in an undergraduate context to use my title, Dr. Coffey. And that seems a bit ridiculous. And it's certainly in the lab, we're on a much less hierarchical situation. But I think that kind of thing is still necessary for women in the classroom. I've also noticed sometimes with collaborators, especially senior male collaborators, there's a bit more of a power dynamic than I have expected from being in the environment that I've worked with people in the past in my PhD. So yeah, I think, and certainly some students and people from cultures in which there is a different gender dynamic, you also sometimes run into situations in that respect where there's just a less respect for women researchers.
Yeah, that's quite unfortunate, but I guess doesn't necessarily come as a surprise, because there's probably, to be realistic, a lot of changes that's go on to a few years before finally removing these kind of like unfortunate instances from the research scientific community. There are more and more researchers that are female, but for sure these kind of hurdles will persist. How when you're confronted to such a situation, how do you overcome them in your behavior or even in your personal life, even if it's something that just happens and that you just kind of think of after and that you realize that was okay, no, that was wrong, that was not normal. How do you overcome these instances?
I think you always second guess yourself and think maybe they're showing a lack of respect because I have done something to make them not respect me, which is always possible. But I think really there's not much to be done aside from continuing to encourage people to be respectful and to treat people according to proper principles in all situations. So I don't really know if I'm dealing with that correctly. Sometimes it does irk me to have had an interaction with someone that I perceive to be unfair or somehow not related to the quality of my work or my background.
Yeah, that's I guess there's probably not a single way to deal with these challenges, and probably different people develop different coping mechanisms with them, and for sure it may affect certain people more than others. But I guess the bottom line is really it's really not something that needs to be changed from the stance of the people receiving that unfair treatment. It's really something that should be changed within the research scientific community as a whole. Although hopefully, like I'm sure that it's less and less with the years. I hope so, but we're definitely far from where we need to be. But
yeah, I think part of how we can cope with it as well is find the good people and work with them. If you're working with someone for a while, you have a good understanding of if there's a problem with that particular issue. And although it sometimes can be painful, you might need to decide that you're not going to further collaborations with people who have different views about gender equality, even if it's subtle. So you find the people that you want to work with, and there are plenty of good people out there. And once you establish that network, you can sort of ignore the rest.
It's a very positive and optimistic way of looking at it. And I'm sure that will help, because if everyone acts that way, eventually, that's going to have an impact on the people that harbor those wrong behaviors. Well, that's it for today. Thank you so much for your time. Congratulations again on your Rising Star Award. Absolutely.
Thank you very much. It's really been a pleasure.